the brain just doesn't develop appropriately. So in the FMR1 gene, we have something called a trinucleotide repeat. Usually there's about 7 to 52 repeats in healthy individuals. But in Fragile X individuals, this repeat becomes expanded to more than 200 repeats. This is very similar to what happens in Huntington's disease, which is also a trinucleotide expansion disease. So this is associated with some craniofacial defects and also mental retardation. In fact, Fragile X is the largest contributor to heritable mental retardation.
So let me see what I said. So it's that you have too many of these genes, or not enough?
No, so it's just this little region outside that flanks the actual gene. This is what would give rise to the protein. And this little repeat just expands. So it's kind of like a control center for the gene. So you can imagine that there's on and off switches upstream. Yeah. And those switches just get all messed up because there's way too many nucleotides there. So it's just a mutation, a very repetitive mutation. And there's a number of diseases that behave this way. And in fact, we've published in the lab a study of Friedreich's ataxia, which is another trinucleotide repeat disorder.
And in healthy individuals, if you say 7 to 52,
so this is a trinucleotide. There's a C, a G, and a G. Your DNA is made up of 4 nucleotides. So this CGG gets, usually there's 7 to 52 of these repeats. But then the disease, it expands to almost 200 or more of those repeats. And like I said, it just kind of messes up the control center for the gene. So eventually you don't get the active form of the protein. So we know a little bit about these diseases. How can we use stem cells to learn enough to have some application? Well, as is evident in the case of Alzheimer's, is that we somewhat have to get back to the basics. And our approach is to derive disease-specific cell lines. And the reason why we want to do this, Jeanne highlighted in her talk, is that pluripotent stem cells can model human development in the dish. So as an example, we'll take those biopsies that Jeanne had mentioned, expand those cells in culture, generate the pluripotent stem cells from the somatic cells or the skin cells, and following that, we can induce those cells to recapitulate neuronal development in the dish and study them at intermediate stages throughout this process. So we can do what we call genome scale characterization. We won't get into the nitty-gritty of this, but we pretty much assay everything that we can assay given the state-of-the-art technology. And we can figure out what exactly is wrong with these cells. Are there mutations? Are they activating genes that they shouldn't? Are they silencing genes that they shouldn't? And at what point in development is this happening? So at this point, once we've established the model, you guys will see this on your tour, we can do we can do drug screening at any stage of this process. You guys are all interested in how fast is this? Once you get the clinical trial going, how long does it take before we can get something actually under works? Well, one thing that we have access to is a chemical library of every FDA-approved drug that there is. So in something like Fragile X, we can differentiate these cells in the presence of all the FDA-approved drugs and see if any of those drugs would prevent the expansion of the repeat. And if so, we wouldn't have to undergo clinical trials, safety trials, and dose dependency trials, and therefore really shorten the amount of time that it would take to get a treatment applicable. And thank you guys very much for your time, and don't hesitate to contact me if you have any questions.